Good morning, children. In this video, we will be talking about mutation and genetic disorder. We will study pedigree analysis, color blindness and sickle cell anemia under the heading of chapter number 5, that is Principles of Inheritance and Variations. This slide shows mutation, a little change, addition or alteration in the DNA, that is mutation. So it is a phenomena which causes alteration of DNA sequences resulting in changes in the genotype and the phenotype of an organism. It leads to variation in DNA in addition to recombination. Loss, that is deletion or gain, insertion or duplication of a segment of DNA results in chromosomes alteration as genes are located on chromosomes. Alteration in chromosomes results in abnormalities. This is known as chromosomal alteration, which are common in the case of cancer cells. Mutation also occurs due to change in a single base pair of DNA. This is called point mutation, and best known example is sickle cell anemia. Deletion and insertion of base pairs of DNA causes frameshift mutations. There are many physical and the chemical factors that induce mutation, which are called mutagens. The best known example of mutagen, that is ultraviolet radiation. This slide shows pedigree analysis. Pedigree analysis is an analysis of traits in the several generations of a family. In this analysis, the inheritance of a particular trait is represented in the family tree over generation. Pedigree analysis or study provides a strong tool in human genetics, which is utilized to trace the inheritance of a specific trait's abnormality or disease. In this, you can see the square, blue color squares, it indicates male, and the red color circles, they indicate female, and red color indicates affected females. So these are the symbols which generally used in the pedigree analysis. Now this slide is of color blindness. It is a recessive sex-linked traits in which eye fails to distinguish red and green colors. The recessive alleles are carried by X chromosome. In females, it appears only when both the chromosomes carry the gene which is responsible for color blindness. The female function as carriers in the presence of single recessive gene. In males, the defect appears in the presence of single recessive gene with the Y, because Y chromosome does not carry any gene for color vision. So if X chromosome, when the zygote forms and X chromosome has recessive gene, then it may appear. Hemophilia and color blindness show cross inheritance pattern, where inheritance of sex-linked characters is transmitted from father to daughter or from mother to son. This slide is all about sickle cell anemia. It is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for a gene, that is heterozygous. This disease is controlled by a single pair of alleles, that is HBA and HBS. Only homozygous individuals for hemoglobin show the diseased phenotype. Heterozygous individuals appear unaffected, but they are carrier of the disease, as there is a 50% chances of transmission of mutant gene to the progeny, leading to sickle cell traits. It is caused by the substitution of glutamic acid by valine in the 6th position of the beta globulin chain of the hemoglobin molecule due to a single base substitution from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle cell structure. So in the next video, we will be talking about the other chromosomal disorders.